Our last speaker is Shinya Yamanaka, and we're especially proud of him at UCSF because he has an appointment not only in Kyoto but also at the Gladstone, and then an appointment as a professor in the Department of Anatomy here at UCSF. Many of you know his work. In fact, probably everybody in the room knows his work on pluripotent stem cells, and it's just been an extraordinary transformation in all of biology that one can take an adult cell and turn it into a stem cell and then turn it back into a differentiated cell. So I'm not going to spend time telling you all about his history. He's going to tell you about the really exciting studies that he's done and continues to do. Shinya?
Thank you, Master. So, thank you very much for your kind introduction, Stan. Good afternoon. I just came from Japan, so I'm still in jet lag. I'm sorry. I spend 95% of my time in Kyoto and 5% in San Francisco. I'm working at Gladstone Institute, but as most of you know, Gladstone has a strong connection with UCSF. Well, Gladstone is located in this Mission Bay campus, and also all the scientists at Gladstone has appointments at UCSF. My appointment at UCSF is professor without salary. So I did my postdoc training at Gladstone 20 years ago. At that time, Gladstone was not here. It was in a very dangerous area, San Francisco General Hospital. Well, 20 years ago, this area, there was nothing except for driving range, golfing. I often came. But the golf range has gone. I'm so sad. So this is my mentor, Tom Inenarity, 20 years ago. The purpose of this slide is to show you that I used to have more hair. So Tom was working on a gene which he thought had a potential to lower cholesterol and prevent atherosclerosis. So he asked me to test his hypothesis. I generated transgenic mice expressing his gene in a liver-specific manner. But it turned out that his gene was an oncogene. We observed this ugly hepatocellular carcinoma by overexpressing his gene. So I then tried to understand why that gene caused these unexpected tumors. So I was able to identify another gene which I thought functioned as a tumor suppressor gene. But it turned out that gene I identified is important embryonic stem cells. When I knocked out that gene, ES cells were fine in proliferation. But when I tried to induce differentiation, knockout ES cells did not differentiate. So that's how and why I got interested in embryonic stem cells. Since then, I have been studying mouse and human embryonic stem cells. So in 1997, I and my family went back to Japan, and I got sick. I suffered from PAD. PAD stands for post-American depression. But thanks to my to supports of my family and of my colleagues and of my friends, I was able to overcome my PhD and continue my science. Then in 2006, we were able to we were able to publish this paper describing iPS cells. 2006 in mouse and 2007 in human. So we showed that by introducing these 4 genes, OX34, SOX2, c-MYC, and KLL4 into somatic cells like skin cells and blood cells, we can reprogram somatic cells back into the embryonic state. ES-like stem cells. So we designated these stem cells induced pluripotent stem cells, iPS cells. Let me show you an animation. I'm sorry this is not as fancy as what Hans just showed, but all we need is blood, and then all we have to do is just add a cocktail of 4 or 5 genes. Then those small number of genes go into blood cells, and these genes initiate the process of reprogramming. So blood cells become iPS cells and they start proliferating. Once they become iPS cells, we can expand iPS cells as much as we want. Then by treating, this is a microscope image of iPS cell generation from blood cells. So we can expand these iPS cells as much as we want. Then by treating these iPS cells with chemicals, we can induce differentiation into many lineages, including beating cardiac myocytes. So when I first saw these beating cells, I remember my own heart synchronized. It happened 6 years ago. So I want to show these 3 people in my lab. It was these 3 people who really generated iPS cells. Kazu Takahashi and Yoshimi Tokuzawa were my very first graduate students, and Tomoko Chisaka was my first research associate. Without these 3 people, we could never have generated iPS cells, at least in our own laboratory. So these 3 people really reprogrammed my life. So with this technology, this is what we are now many people are now trying to achieve. We want to apply this technology in medicine, in cell therapy, in toxicology testing, and also in drug screening. So let me begin with cell therapy. The Japanese government has been spending a lot of money to promote regenerative medicine using iPS cells. So we're hoping that these applications will go to clinic within the next 3 or 4 years. Macular degeneration, Dr. Masayo Takahashi in Kobe, she got an approval from the Japanese government already. So I'm sure she will start the very first clinical trial sometimes next year. Other applications will follow within 3 or 4 years. But I really think the real potential of iPS cells is not in cell therapy. Rather, it's in drug screening, in toxicology testing and making disease models and drug screening. I believe that's the best applications of this technology. So as you know, many drugs could have this kind of side effects in heart. They could cause lethal arrhythmia. If this happens, pharmaceutical companies have to withdraw their drugs from markets. It's a huge loss to the company and also to patients. They really want to predict this kind of cardiac toxicity as quickly as possible. However, it is almost impossible to obtain a good amount of human cardiac cells for such kind of safety testing. So instead, pharmaceutical companies have been using tumor cells, like CHO cells or 293 cells. In order to make those tumor cells a little bit cardiac-like, they introduce just one gene, one cDNA, a hERG cDNA encoding potassium channel. So this is, of course, very artificial system. And I'm sure this test suffers from both false positive and also false negative. But now we can use beating cardiac myocytes derived from human iPS cells. So we hope that by using this system, the prediction of cardiac toxicity should become much more sensitive and much more accurate. Well, one of my colleagues, Dr. Yoshinori Yoshida, has been testing this kind of multi-electrode array system to have this kind of in vitro in dish ECG. So we are now trying to bring this technology to companies. How about making disease models and drug screening? Well, let me use ALS as an example of this application. As you know, ALS is well known as Lou Gehrig disease in this country. So this disease has been around for more than 100 years, but we still don't have good cures, good medicine for these patients. In these patients, motor neurons get degenerated and eventually die. So patients lose their ability to move. However, they can still think, they can still sense. So it's a very tough condition, not only for patients, but also for family members. Many scientists in the world, including my colleague, Dr. Haruhisa Inoue, has been applying iPSO technology to understand ALS and to search for effective drugs. So Haruhisa has generated iPS cells from both control individuals and ALS patients. When iPS cells are undifferentiated, we did not see any differences. But as soon as Haruhisa induced differentiation of iPS cells into motor neurons, he observed marked differences in many ways. The typical difference is in morphology. As you can see, motor neurons from ALS patients have much shorter neurites projections. So they only have like 50% of neurite length. So we are now using this phenotype as to screen thousands of chemicals. We have set up this kind of mini robotic system. And Haruhisa has identified several chemicals which can revert the abnormal phenotype of ALS motor neurons back into the normal phenotype. So we're hoping that some of these chemicals turn out to be effective in real human patients. So, ALS is relatively rare diseases, but I believe we can apply the same technology to more common diseases, including like Alzheimer's diseases. And we hope that by utilizing iPS cells from each patient, we can establish we can advance personalized medicine. This is what we are now doing. When we have a good drug, we treat all the patients with the same drug. We just give the drug to all patients. This drug may be effective on some patients, but not effective on others. And on some patients, this drug may give some bad side effects. So again, Haruhisa has generated iPS cells from many Alzheimer patients. Here we only show the results of 4 patients. We have 2 familial cases, different mutations in APP gene, and we have 2 sporadic patients. When we generated neurons and astrocytes from these 4 ALS patients, not ALS, I'm sorry, Alzheimer patients, we observed marked differences. In one familial case, APP E693 delta, and in one sporadic case, we observed intracellular deposit of A-beta oligomers in neurons and also in astrocytes. In these 2 patients, we observed cellular stress caused by A-beta oligomers. And also in these 2 patients, we found that neurons can be protected by DHA treatment, but not in the other 2 patients. So this simple experiment clearly showed that each Alzheimer patient is different. For some patients, some drug can be effective, but not for others. So by performing this kind of iPS cell testing, I hope in the near future we can do this kind of personalized medicine. So for some patients, we will give drug A. For other patients, we will give drug B. For other patients, we should use drug C. Well, very similar issue can be applied to clinical trial for new drugs. So we want to do kind of selected clinical trials with iPS cell technology. This is conventional clinical trial. We apply the same drug candidate A, to all patients. In some patients, drug candidate A can be effective, but in other patients, candidate A are not effective, and in other patients, drug A, candidate A may cause some side effects. So this is what we are now trying to do. Clinical trial in dish. We are now trying to generate many iPS cell lines from many people. And each well of this dish has iPS cells from individual. I mean, like neural cells, neurons from iPS cells. And we want to test the effect of drug candidate in each patient in Petri dish. Then we can select individuals which are likely to show effects to drug candidate A. So we just select those individuals for the real or selected clinical trial. So as you know, pharmaceutical companies have been spending billions of money in drug discovery. And I believe many good drugs may have been discarded because of such kind of conventional clinical trials or false positive in hERG test. So I hope that by applying iPSC technologies, we may be able to rescue some of those potentially good drugs. So again, I really think that the real potential of iPS cells is in drug discovery rather than in regenerative medicine. So finally, I want to thank my colleagues in Kyoto. We have more than 300 people in this building, Center for iPS Cell Research and Application. This is only a part of those people. You know, at Gladstone Institute, I also have a group. It's here. I have a small but very great people. So with this, I'd like to finish my talk, and finally, I'd like to thank the Foundation, Breakthrough Prize Foundation, and UCSF for giving me this great opportunity. Thank you very much.
Okay, questions? Wait, use the microphone. Thanks.
I was a little bit surprised that you don't seem to be so optimistic about using the iPS cells for regenerative medicine, etc. I thought that this was, some people think this is probably one of the most intriguing applications. Is there any particular reason why you're not so excited about that?
Well, I'm sorry, I am excited about but, you know, especially in Japan, there have been a huge support from the government. So it's going very well. But as for the other application in drug discovery, I believe we could do much better. We should do much better job. So that's why I want to stress the importance of this application.
Other questions?
Is it possible that the epigenetic marks that iPS cells acquire and ultimately for use in regenerative medicine, for example, might be incompatible with fully normal differentiation in the tissue types where you'd like to apply these cells?
Well, so it's been controversial. You know, some reports said that iPS cells maintain somatic memories, like iPS cells derived from blood cells can differentiate back into blood very well, but not into like neurons. But we have been testing many iPS cell lines, and we found that when reprogramming is not complete, we do see such kind of somatic memory. However, when reprogramming is nearly complete, iPS cells are very similar to embryonic stem cells. So I think we still have to see, but I think we can overcome such kind of epigenetic memories in many applications, if not all.
Other questions? Let me ask one about how you're very interested in drugs and screening compounds, and what a rare one might turn into a drug. Can you produce huge quantities now of these cells? Typically, in screening, you need cells that double in 24 hours to do this. Are you able to do this now? Because we've had trouble trying to do that.
Well, well, some cells, we could do that. The advantage of iPS cells is that we can expand iPS cells as much as we want. So the issue is differentiation. Well, we still need some, progress in that aspect.
All right, and then in closing, I want to thank you again.
Thank you very much.